neuroscience is really important because it's the search for the truth. Knowing why and knowing how is the necessary first step to really doing something about solving a problem. I'm very privileged to have the chance to take care of patients and also run a research laboratory. It's fascinating to probe into nature, but also have a direct patient impact. So my lab is focused on the non-coding genome. All the cells in the human body carry the same DNA, but your skin cell is not the same as your heart cell or your brain cell. So these cells have to make choices about which of the genes to actually express. This non-coding genome is involved in these choices. These are DNA switches that decide when and where genes turn on and off. So our research is really about what information the cell chooses to make use of. And that choice has a lot of implications for how cells behave, whether they're healthy or not, and therefore impact on human disease.
So we're just looking at HER2 expression levels in this experiment?
My clinical experience informs the kind of questions and technologies we use in the lab and what problems to pursue. One of these questions was why 4 out of 5 patients with autoimmune diseases are women. In the past, people thought it would be due to hormones. We discovered that it has to do with the different chromosomes. Women have 2 X chromosomes, whereas men have an X and a Y chromosome. And there's a very special RNA that's only made on the 2nd X chromosome. And that RNA we found is responsible for this sex-biased immunity. Genome science is at a very interesting inflection point. We've moved from technologies for reading the information to writing or changing information. The switches are still there, but we can flip them on or off at will. This is a tremendous area of investigation, especially in cancer. Using our technology, we discover that there's a quite common situation where a cancer-causing gene will break off from chromosomes and turn into its own separate element, a ring-shaped circular piece of DNA called extrachromosomal DNA. We learned that in extrachromosomal DNA, all the switches get flipped onto the on position. And in these extrachromosomal DNAs, cancer cells gain an advantage in gene expression. Every molecule of extrachromosomal DNA makes 4 times the messenger RNA than the exact same sequence on the chromosomes. So imagine if you're in an argument with somebody and they talk 4 times more, they would be winning the argument. And that is what the cancer cell is doing. But we found that that process of making RNA all the time comes with a cost. And we can exploit that conflict in making RNA all the time and try to copy DNA and create a drug that makes these kind of cancer cells self-destruct. That drug has now moved into clinical trials in patients. I love what I'm doing. It's so gratifying for me to see the methods that we developed being used by scientists around the world to advance their own programs.